start by talking about the cell membrane, right? Why are we okay? So, when we're talking about the cell membrane, if you remember when we did the topic of cell biology and all that, I had specified that the topic of cell membrane we have in detail in our syllabus. So, we will go over the topic then. So, we have covered nearly all of the parts of the cell, all of the organelles of the cell beforehand. Today, we will take a look at the cell membrane. We are first going to go ahead and start by talking about what is the structure of a cell membrane. Okay, how is a cell membrane made? And once we know what is the structure of a cell membrane, we are going to be able to better understand the properties, right? How does a cell membrane function the way it functions, right? How does a cell membrane actually do its job? And how does a cell membrane helps in transport of molecules from in the cell to out the cell or out the cell to in the cell, right? That's the job of the cell membrane. It's regulating, it is controlling who is coming into the cell and who is going out the cell. So before we can actually understand how is it that the cell is doing something like that, how is it that a cell is actually going over a concept like that, we need to understand how a cell membrane actually looks like, or what's the actual structure of it. So when we talk about our cell membrane, before we can understand about our cell membrane, we need to understand something, a type of lipid. Okay. Structure of cell membrane. This is what we are starting with. When we talk about, so cell membranes are made up of a structure that is known as phospholipid. Okay. Phospholipid in terms of structure are similar to the lipids that we studied in biological molecule. What did we study in lipids? We studied in lipids that generally lipids are formed because of the reaction, condensation reaction between glycerol. Glycerol was our alcohol and fatty acid chain, right? And the bond that we form between the glycerol and the fatty acid chain is our ester bond. Now, phospholipid is exactly the same, but it also has a phosphate group attached to it. So along with glycerol, along with fatty acid, it also has a phosphate group attached to it. Now, why is all of this important? Why are we talking about all of this? Now, your structure sort of looks like this, okay? The structure is going to be looking like this. This over here is my molecule of glycerol, okay? Attached to the molecule of glycerol, if you remember, I had mentioned that we're going to have our fatty acid chains. So attached to this molecule of glycerol, I will have two fatty acid chains. Okay. In triglycerides, if you remember, I told you triglycerides are the commonest type of, the most common type of lipids that we have. So in triglycerides, there was one glycerol and three fatty acids. In phospholipid, we have one glycerol, two fatty acid, and then we have our phosphate group. Right? Now, the question that's going to come is that why are we studying about this phosphate group? What is this phosphate group? How is this phosphate group affecting our molecule? How is this phosphate group affecting our molecule? So this phosphate group affects the molecule in such a way that it changes certain properties of the molecule. So this is the phosphate group. We call it the phosphate head. Okay. Over here, I have my glycerol. And then I have my fatty acid chain. Now, we were discussing about why is it that this structure is important, or why is it that having a phosphate group is changing the property of the lipids? Shouldn't it work like the lipids we have already studied? Now, because it has a phosphate group attached to it, the properties are slightly different. How are the properties slightly different? If you remember, when we did the topic of our previously, when we did the topic of biological molecules, I told you that lipids are generally insoluble in water, right? Lipids don't dissolve in water. Fats don't dissolve in water. Oil and water will never dissolve. So fats generally, lipids do not generally dissolve in water. That's what I told you. But that's not 100% true for phospholipids. Why? Because phospholipids have the phosphate group and they have the fatty acid chains. Now, the fatty acid chains are going to be acting as hydrophobic region, right? The fatty acid chains are going to be acting as the hydrophobic region. In fact, we call them hydrophobic tails, right? The fatty acid region of the molecule is going to be completely hydrophobic. It's going to be completely water hating. That means that the hydrophobic tails do not want to be close to each other. They don't want to be close to water. They want to be as far away from any aqueous solution at all cost, right? So these tails are hydrophobic. On the other hand, the phosphate group that we are seeing, this phosphate group is hydrophilic. It loves to be in the water. It loves to be surrounded by an aqueous solution, right? So the hydrophilic head wants to be in the water. While our hydrophobic tails are water hating, they want to be as far away from the water as possible. And of course, we're going to have the glycerol in the middle. This is the glycerol. Now, what's going to happen? How is it that a phospholipid is going to be able to make a cell membrane? Because a phospholipid has a region that wants to be in the water, and then it has a region that wants to be away from the water. So our cell membrane is actually, we use a term for it. The term we use for the common cell membrane we see in our body is that we call them a lipid bilayer. That's what we call them. We call them a lipid bilayer. Why do we call them a lipid bilayer? So, the reason we call it a lipid bilayer is because, first of all, there are two layers of phospholipids. Okay? Two layers of phospholipids that are going to be present. So, we have two layers of phospholipids. That's why, first of all, it's a bilayer because we have two layers. Now, how is it that two layers of phospholipid are able to arrange themselves in such a way that they can allow a cell to function? Because this molecule has both parts that want to be in the water and parts that hate the water. How do they find that balance? So they arrange themselves in a very certain way. They arrange themselves in such a way that the hydrophilic portion of it, the portion that likes to be in the water, is going to be facing the side that is in the water, right? For example, the cytoplasm of the cell. So the side of the cell membrane that faces the cytoplasm will have the hydrophilic region, right? Then we have the hydrophobic tails. The hydrophobic tails will all arrange themselves in such a way that they all are pointing towards each other. Like so. Alhamdulillah, this is the first layer. Alhamdulillah. This is the first layer and this is the second layer of phospholipids. Okay. So as you can see, it's a bilayer because it's made up of two layers of phospholipids. So we are going to be considering it a bilayer. And of course, what's going to be happening? The phospholipids are going to be pointing towards the center. Why is it that the phospholipids, these fatty acid chains are all pointing towards the center? Why are they pointing towards each other? Because by pointing towards each other, they create a central region that is hydrophobic. This this center region of the cell membrane, the middle part of the cell membrane is hydrophobic. So the fatty acid chains are all pointing towards each other. So they sort of create an area where there is no water. By pointing towards each other, this create an area where there is no water present. So this area becomes hydrophobic. And since there is no water present, because all the water-hating molecules have come together, so what is happening? This area has no water in it, so the hydrophobic portion can exist quite happily. Now, we have the hydrophilic heads, right? The hydrophilic heads, what is going to be happening? The hydrophilic heads are going to be facing the fluid that is on the outside or inside of the cell. For example, inside of the cell, we have cytoplasm, right? So this side, the hydrophilic side, is going to be facing your cytoplasm. Outside the cell, we have tissue fluid mostly. Or for example, if it's a WBC, if it's a red blood cell, then there is blood or there is tissue fluid or whatever, right? Whatever fluid is found outside, that is present on the outside of the cell. So a cell membrane is made up of two layers. We call it a lipid bilayer, okay? We will have an outer layer and an inner layer. The outer layer is going to be arranged in such a way that the hydrophilic head is facing the tissue fluid. The inner layer is facing in such a arranged in such a way the hydrophilic head are facing the cytoplasm. The hydrophobic tails of both the layers will be pointing towards each other, and as a result, the center region will become completely hydrophobic, water hating. At the same time, because I have the hydrophilic region outside, right? Because I have the hydrophilic region outside, what is going to end up happening? That the cell is still able to survive in fluid. The cell is still able to exist within the blood. It's still able to exist within the fluids of the body because the outer layer is hydrophilic and the innermost layer is also hydrophilic and the center region is hydrophobic. So the cytoplasm, tissue fluid, cell membrane can exist within all of this. So this is the basic structure of what a cell membrane looks like. This is not the full structure of what a cell membrane looks like, but this is the basic structure of what a cell membrane looks like. We call it a lipid bilayer. Now, phospholipids don't always just make a lipid bilayer. It's not that every single time they are only going to be making the lipid bilayer. That's it, right? They are also, if you're going to go ahead and talk about it, they are also make another kind of structure that we call micelles. Okay, micelles, an easy way to understand micelles is sort of like a vesicle. Think of it that way, right? What happens is this, that if I take my phospholipid and I put it in water and I shake them all up. If I take my phospholipids, I put them in water and I shake it all up, I will notice something quite interesting. I will notice, the interesting thing that I'm going to notice is this, that my phospholipids are going to arrange themselves in a very unique way. The phospholipids will arrange themselves in such a way that they will end up making a spherical structure like this. So if I just take some random phospholipids, right? I'm not making the cell membrane. If I just take some random phospholipids, so what am I going to observe? That the phospholipids are going to arrange themselves in such a way within water, right? I've mixed them with water. They're going to arrange themselves in such a way that they will end up making a structure that we call a missile. What is a missile? A missile is, like you can see, it's a sphere. It's specifically a sphere that has the hydrophobic region facing the inside and the hydrophilic region facing the outside. This is the nature of phospholipids. They will always try to arrange themselves in such a way that the hydrophilic region is facing the water and the hydrophobic region are all facing each other. So in our cell, this is how they work in the form of cell membrane. But if you just take some phospholipids and mix them with some water, you will automatically see that the phospholipids are arranging themselves in this particular shape. They're arranging themselves in a sphere in such a way that the hydrophilic tails are all pointing towards each other. Sorry, hydrophobic tails are all pointing towards each other. And the hydrophilic region is the one that is actually facing outside of the water. So this type of a structure is what we call a missile. So phospholipids can either give you a structure like this, or they will give you a lipid bilayer, which is phospholipid bilayer, we call it, which is actually what we observe in our cell membrane. In our cell membrane, we actually see it as a phospholipid bilayer. So this is a very, very, very basic structure of our cell membrane. Any confusion so far? Does anybody have any questions to ask? No. Are you sure? All clear? No confusion in this part? Aruch, is everything clear? Yes. No confusion? Okay. Now, when we go ahead and we talk about it, so this actually is a basic structure of the cell membrane. Why is this a basic structure of the cell membrane? Because our cell membrane isn't just composed of phospholipids. It's composed of a lot of different things mixed together. For example, we have a lot of different kinds of proteins, right? We have a lot of different kind of proteins that we are going to be seeing in our cell membrane. What are they? What is the actual structure of the cell membrane? So the actual structure of the cell membrane, we have a term for it. We call it the fluid mosaic model. Why do we call it the fluid mosaic model? The reason we call it the fluid mosaic model is because, one, that it is fluid. As you know, cell membrane is not a rigid structure, right? Cell membrane is a fluid structure. The membrane has to be fluid for the cell to be able to do a lot of its function. For example, red blood cells need to squeeze through small capillaries. Your white blood cells need to squeeze through the capillaries to get to the site of infection. Wherever the infection is, they have to get there. So they have to squeeze through the capillaries. Cell membrane has to be pretty fluid. You have exocytosis, endocytosis. We will study that later on. So it has to be fluid. Mosaic. Mosaic, if you remember, when you were a kid, you might have done, in your arts and craft activity, you might have done activities where you would take a piece of paper, you would cut it into small pieces, and then you would join them together to make a picture, right? That is mosaic art. When you make a picture using smaller pieces, that's a mosaic art. So mosaic, because all of the cell membrane components are fitting together in the same manner. All of the cell membrane components are different sizes. They look different, but they all fit together to give you a mosaic-like picture, to give you a mosaic-like cell membrane. So the cell membrane actually follows what we call a fluid mosaic model. So fluid mosaic, when you talk about your cell membrane is visible using the electron microscope. Okay, not the light microscope. The electron microscope is the one that allows you to actually see the cell membrane, right? And it is around 7 nanometers in width, right? If you take a look into it, they are around 7 nanometers in width. Now, when you go ahead and you talk about it, when you go ahead and you talk about it, so what are the structures that are going to be found in the cell membrane? Right, because I told you it's a mosaic. We have got different components mixed together to give you a cell membrane. The first component we have already studied quite well. That is the phospholipid bilayer. So the phospholipid bilayer is the first component that you will find within the cell membrane. Now, along with that, you are also going to find proteins. We are going to study about two types of proteins. We are going to study about, in proteins, we have two types of proteins. The two types of proteins that we have is one, we have intrinsic proteins, or we like to call them integral proteins. And the second feature we have is that we have extrinsic proteins. We have extrinsic proteins, okay? And extrinsic proteins, which we also call peripheral proteins. What else? What else will we find in the cell membrane? So we have phospholipids, we have our proteins. Another really important thing that we find in our cell membrane is cholesterol. Right now, I'm just going over the structures we will find, and I will just discuss with you guys where are they found, how are they found, and how do they actually work. So we will find our proteins. We will find structures such as glycolipid. We will find glycoproteins. We are going to find glycoproteins, glycolipids. These kind of structures are going to be found as well within the cell membrane. And when you're going to go ahead and talk about it, so these are the different type of components that you are going to be finding within your cell membrane. Now let's go ahead and actually start by discussing each of these points individually. So we know, if you remember, we said that the first thing that we're going to be seeing is going to be a phospholipid bilayer. So that's the first thing. We have talked about what is a phospholipid bilayer. We have discussed that a phospholipid bilayer, is basically your phosphate groups are going to be acting as the hydrophilic head, right? And they're going to be facing the outside of the water, right? And the phospholipid fatty acid chain will be facing the inside of the cell membrane, creating a hydrophobic center. Right? Creating a hydrophobic center between the two layers. Then, of course, like I said, that the phospholipid heads are going to face the aqueous medium, right? The phospholipid heads will face the aqueous medium. Now, when we talk about our fatty acid chain, right? The fatty acid chain can be saturated or it can be unsaturated. What do we mean by saturated or unsaturated? The exact same thing that you guys might have studied in your chemistry. In chemistry, we studied that saturated hydrocarbons are the ones that only have single covalent bond. Unsaturated hydrocarbons are the ones that have a double covalent bond. So, phospholipids, similarly, the fatty acid chain can either have only single covalent bonds or it can have double covalent bonds. So, if it has double covalent bonds, we call them unsaturated. The more unsaturated the fatty acid, the more fluid your membrane. This is one of the factors. One of the factors that affects the fluidity of the membrane is our saturation. So the less saturated, more unsaturated fatty acids are going to be. So the more fluid your membrane is going to be. Why? How does saturation, what does double bonds have to do with our fluidity? Because when they're going to be unsaturated, so the fatty acid tails are often bent, right? They are often bent because of the double bond that exists between them. It changes the shape of the molecule, and you and I have studied in chemistry shapes of molecules, so we know that the how the bonds change the shape of the molecule. So it will go ahead and it will change the shape of the molecule. So the tails are bent. As a result, the tails fit together loosely, right? Instead of being tightly packed with each other, the tails are fitting together quite loosely with each other. So that is going to make our cell membrane more fluid, which is good. That's what we want. We want our cell membrane to be fluid, right? Now, along with that, what are we going to be seeing? That fluidity is affected, one, by how unsaturated the fatty acid is. Two, fluidity also depends upon the tail length. It also depends upon the tail length, right? The longer the tail, the less fluid the membrane. So the longer your fatty acid chain, the longer the tail, the less fluid our membrane is going to be, which is again good to a certain extent. We will discuss this in a bit more detail, but we don't want the membrane to be too fluid, because if the membrane is too fluid, then how are we going to maintain the structure of the cell? But at the same time, we don't want the cell membrane to be too rigid. So we want it to be in the middle. We want it to be moderately fluid. So fluidity will also depend upon the length of the carboxylic acid chains. Fatty acids are carboxylic acids. So what's going to happen? So that is the first structure that we have in our membrane. Now, the second structure that we have in our membrane, and that is important for us, is proteins. Okay. When we are going to talk about proteins, so I told you that your proteins are basically of two types, right? We have integral proteins and we have intrinsic proteins and extrinsic proteins. To understand this, let me first draw the structure of the phospholipid membrane for you, right? Let me go ahead and actually go over the structure of the phospholipid membrane with you. Now, what is going to be the structure of the phospholipid membrane? The structure of the phospholipid membrane is going to involve, first of all, we have the phospholipid bilayer, right? So let's start by drawing that. Along with our phospholipid bilayer, we are going to have proteins. Now, I mentioned that we have two types of proteins. We have intrinsic proteins and we have extrinsic proteins. First of all, what's the difference between the two of them? Why are we talking about them separately? So, intrinsic proteins are proteins that generally exist on the inner side of the cell membrane. right? They generally exist on the inner side of the cell membrane. What do I mean by that? Let's say that I have a protein that is spanning or that is existing the entire length of the cell membrane. This is if you go ahead and talk about it, this is an example of an intrinsic protein because this protein is existing on the inner surface of the cell membrane. So we would call it an intrinsic protein, right? Um, we would call it a intrinsic protein because it is found embedded within the membrane. We find them embedded within the membrane. So we call them intrinsic proteins. Now, it's not necessary that intrinsic protein would only be present throughout the cell membrane, right? Intrinsic proteins can exist like this as well. Once intrinsic proteins, it's not necessary that they're going to be spread throughout the entire cell membrane, right? That they will go from one layer to the other. You can also have intrinsic proteins, something like this, but it's within the cell layer, right? So intrinsic proteins are generally proteins that exist within the cell layer, right? As long as they're embedded within the cell layer, we call them intrinsic proteins. Now, so what are extrinsic proteins? I think now you'll be able to guess quite easily what extrinsic proteins are. Extrinsic proteins are proteins that are going to be facing the outside of the cell membrane, right? So extrinsic proteins are proteins that would be facing the outside of the cell membrane. How is that going to look like? That is going to look something like this, right? This would be an example of what? This is an example of your extrinsic protein because as you can see, this protein is existing on the inner surface of the cell membrane, right? It's existing in the inner surface, I'm saying inner surface, sorry, the outer surface of the cell membrane. So this kind of a protein is going to be known as an extrinsic protein, right? So we have two types of proteins that we have to go ahead and talk about. So we can either have, like I said, we can have our, we can go ahead and we can have intrinsic proteins, or we can go ahead and we can have our extrinsic proteins. Now, when we are talking about our intrinsic and extrinsic proteins, like I said, it's not necessary that our intrinsic protein is going to only be spread throughout the cell membrane. This intrinsic protein, by the way, can also be like this, right? As long as it is present within the cell membrane. It can also be like this. It can also exist on the lower membrane. So over here, I drew it on the inner membrane just to clarify the concept. It can also exist over here on the inner membrane, right? Both the upper layer and the lower layer can have the intrinsic protein. The point is that it needs to be facing the inside of the cell membrane. That's the criteria. Then we will have those that are facing the outside. The ones that are facing the outside, we call them extrinsic proteins. So now if you take a look, can you see why we call it a mosaic? Because you literally have these pieces that are sort of fitting together to give you the cell membrane, different size pieces that are fitting together to give you the cell membrane, right? So now let's actually discuss in detail about these proteins, their function, how do they act, what do they do. Let's go ahead and go with that. Now, let's start by first talking about our intrinsic protein. Now, intrinsic proteins, like I told you, are embedded within the cell membrane, right? They're embedded within the cell membrane, and you can find them in the inner layer or outer layer or spanning the entire length of the cell membrane. So through the entire length, through the entire width of the cell membrane, you can find the protein as well. When it is spanning the entire length of the cell membrane, when it's going from one end of the cell membrane to the other, that type of protein is specifically, we use a term for it. It's still an intrinsic protein, but we use a specific term for it. We call it a transmembrane protein. Why do we call it a transmembrane protein? Because it's going through the entire membrane. It's starting from one layer and going all the way to the other. So we call them transmembrane proteins. Why is it important that we talk about these transmembrane proteins, or we go ahead and we talk about these proteins? Now, intrinsic proteins have both hydrophobic and hydrophilic regions. They have both. They will have a region that's going to be hydrophobic, and they're going to have a region that is hydrophilic. How is it that they're able to stay within the cell membrane? You and I know that the inner surface of the inner region of the cell membrane is hydrophobic, right? So if the inner region of the cell membrane is hydrophobic, then how is it exactly that these proteins are able to exist? The proteins have to also be hydrophobic. How can proteins be hydrophobic? Amino acids. Remember we studied amino acids? Biological molecules. So the proteins or the amino acids will have hydrophobic R group. They will have hydrophobic R group, which is why the the proteins are able to exist within the cell membrane, right? So the amino acids will have hydrophobic R group facing the hydrophobic chain, the fatty acid chain, right? So as a result, the molecule, the protein is able to exist within the cell membrane. Otherwise, it would not be able to, right? Then when we talk about the hydrophilic region, the hydrophilic region is going to be facing either the outside of the cell or the inside of the cell. Or if I go ahead and talk about it, there are a lot of these transmembrane proteins, especially, they act like your they act like your transport protein, right? A lot of these intrinsic proteins act like transport proteins. How do they act like transport proteins? Let me drop it. We have our phospholipids, and then I have my intrinsic protein, like so. Okay. Now, what happens is this, specifically speaking, what happens is this, that the the portion of the second. Okay. The portion of this particular molecule, so that is facing the fatty acid area, this portion, for example, this portion would be made up of hydrophobic amino acids. Okay. This portion would be made up of hydrophobic amino acid. so that this molecule can actually exist within the in the cell membrane. Now, a lot of these molecules actually act like transport molecules, or they act like channels, right? Meaning that they will have a center region that's going to be that's going to allow for transport to take place. They will allow for substances to go in from the one side and come out the other, right? So a lot of these act as transport molecules. So what's going to happen, that the side of the molecule that is facing the outside, or the side of the molecule that is making the channel, these sides will all have hydrophilic amino acids, right? This is what the structure is basically going to look like. This is one of the functions of of these proteins that we have. We will do more functions in more detail. So that is one thing I'll just go over with you guys in a second. But so this is what we were discussing, that our this is what we were discussing, that the hydrophilic amino acids will face the aqueous side of the cell, aqueous side, or the inner side of the transport channel. So if it's acting like a transport channel, then you will find it on the inner side of the channel as well. So this is how your proteins are able to allow the diffusion of molecules. This is how proteins are able to allow diffusion of molecules, transport of substances, but at the same time exist in that hydrophobic environment, still exist within that hydrophobic cell membrane. Now, continuing about what we have to discuss about our molecules, our proteins. So, when you talk about our proteins, a second type of protein that we have is our extrinsic proteins, right? Now, when we talk about extrinsic proteins are found either only on the inner or outer surface of the cell membrane. You won't find them within the cell membrane. They're not within the cell membrane. They're going to be either in the inner or the outer surface of the cell membrane. So you'll either find them on the inner or the outer surface of the cell membrane, right? Now, when we go ahead and we talk about it, there's not much that we have to talk about in terms of intrinsic protein. One thing that we have to talk about now, in your syllabus, most of the topic is related to intrinsic proteins. In your syllabus, you don't need to know extrinsic proteins in any greater detail. This is enough information for you to know what in extrinsic proteins are. Now, many of the proteins are attached to, many of the proteins are attached to your um, carbohydrate chains. A lot of these proteins are attached to carbohydrate chains. These proteins that have a small carbohydrate chain attached to them, we call them glycoproteins. Glycoproteins. Proteins because they are the main structure is protein, but since they have a small carbohydrate chain, we call them glycoprotein, right? Similarly, if you're going to go ahead and talk about it, if we have lipids, right? So if we have lipids with a small carbohydrate chain, we call them glycolipids because the main structure is lipids, but then they have a small carbohydrate component, so we call them glycolipids. And we will take a look when we go over the different components of cell membrane and what is the role of the components in the formation of cell membrane. We will go ahead and we will discuss why are glycoproteins and why are glycolipids important for all of us? Why are we talking about all of these in so much detail? Another really important structure that I want mention to you guys was, of course, that we have, one second, was of course that we have our cholesterol molecules, right? Because phospholipids, proteins, and cholesterols were what we have. Okay, this is what it looks like. So if you have a cell membrane, and on that cell membrane, you see chains like these. This is your protein, and on the protein, you see small chains like this. And these are chains of carbohydrate, right? So we call them glycoproteins. And we will go over the roles of glycoproteins. Why are glycoproteins important? So whenever we have a small carbohydrate chain, so that is going to be known as a glycoprotein. This structure is going to be known as a glycoprotein. Now, along with that, we have cholesterol. The molecule of cholesterol, you will literally see it will be spaced out throughout between these cells. Throughout between these cells, sorry, I said cell. I mean between the phospholipids, in different places, you will see molecules of cholesterol. Why do we have these different molecules of cholesterol spread throughout the entire region? Now, the main reason we will discuss when we do the different roles of cell components, I will tell you the exact way how it happens. But the major reason why we have these cholesterol molecules is because they help maintain the fluidity of the membrane. They help maintain the fluidity of the membrane in such a way that the membrane is going to be it's going to make sure that the membrane's fluidity is not affected, right? Meaning that the membrane is going to continue to maintain that length, to maintain that fluidity within a certain limit. I told you guys it's very important to maintain the fluidity of the membrane in a certain limit, right? We have to maintain the fluidity of the membrane within a certain limit. So we are able to do that. We are actually able to do that because of cholesterol molecules. The cholesterol molecules make it so that when the temperature is too high, the membrane will not become too fluid, because if the temperature is too high, the cell membrane can become too fluid. It can become too loose. And then that's bad because I'm not maintaining the integrity of my cell. Then the other problem that we can have is that we can have another problem that we can have is that if the temperature is too low, the cell membrane can become too hard, too rigid, which is again a problem. So in order to avoid all of that, we need the help of our cholesterol molecules. Okay, another thing that I mentioned was that we have glycolipids. So if I have a lipid, right? For example, over here, this is my phospholipid. And on top of the phospholipid, we have a carbohydrate chain. So this is going to be known as a glycolipid. And glycolipids and glycoproteins have important roles that we will take a look at what they actually are. Now, some of these transmembrane proteins can actually act as channels, or they can act as transport protein. Okay. Now we can have different types of proteins. That is also something we will take a look at in this chapter. So they can either act, as like channels, or they can act like, we will talk about carriers. They can act as carriers. They can act as your pumps that we need for active transport. So these integral proteins generally help in transport. So they can act as a carrier or they can act as a channel. The point is that they will be helping us in the formation, in the actual transport that is going to be taking place, right? So this is actually what the structure of our cell membrane is. So now, and if you now take a look, I think you guys can see quite clearly why we are calling this a phospholipid bilayer, right? We call it a phospholipid bilayer, or why do we call it a mosaic model? We call it a mosaic model because all of this looks together like pieces we have joined together to give us a cell membrane. Now, when we talk about all of this, let's discuss a few of the roles, right? Why, for example, okay, let's start with phospholipids. Why do we need phospholipids? For what do we need phospholipids? What is the role of them? Why do we need them in the cell membrane? Why does it have to be made in such a complex manner? Phosphohydrophilic, hydrophobic. Why go through all of this hassle? Phospholipids, now remember this function of a cell membrane is to regulate the transport or exchange of substances. We don't want substances to be able to leave our cell whenever they want. I don't want any molecule to be able to enter into my cell whenever it wants to. My cell has to be controlled. There are important chemical reactions taking place inside the cell. If I, for example, I can't just let the molecule of glucose leave the cell. I need it for respiration. I need it to stay within the cell. So the cell membrane is the one that actually controls what is happening, who is going into the cell, who is going out of the cell. It's like a border, right? The border of your country. Now, how does it do that? Because of the phospholipids. The phospholipids create and create a hydrophobic interior between the layers. Between the layers we have in hydrophobic region, right? So polar molecules, when you have your polar molecules or ions or water soluble molecules, right? Such as sugar or such as your amino acids or such as your proteins cannot cross the cell membrane. If they want to go across the cell membrane, for that, they will need to go through the transport proteins that I was telling you about, right? They cannot directly just move in and out of the cell. They need to go through transport proteins. And transport proteins are not easy to go through, right? Some of them require ATP. Some of them require energy. Some of them will only work in a certain way. So all your polar molecules, water soluble molecules, large molecules, really big molecules, they are not going to be able to enter into the cell, right? Along with that, your non-polar molecules, small non-polar molecules like oxygen, like carbon dioxide, can freely move across. We don't want water, oxygen to be, to depend upon the cell membrane to come in and out. It's small, it's really small in size. So it can easily cross the cell membrane and it's non-polar. Polar molecules, on the other hand, will not be able to cross. And large molecules also, even if it's non-polar, but it's large in size, it cannot directly cross the cell membrane, right? So this is how the cell membrane helps in controlling what is happening inside the cell. Along with that, a lot of the phospholipids can act as signaling molecules. What do we mean by signaling molecules? We're going to study, inshallah, cell signaling later in this chapter. But signaling molecules is that they can signal the other cells, right? They can send signals to the other cells. So as a result, they can activate other molecules. For example, they can activate enzymes, right? We are going to study cell signaling, inshallah, in this topic. They can activate enzymes. They can signal the enzymes that, listen, you're needed over here for some kind of action, for something that we need to be, for something that need to be done, right? Another thing that we can do is that we can hydrolyze means breakdown. We can hydrolyze phospholipids, and after hydrolysis of phospholipids, we can release small water soluble glycerol type molecules, right? They're not exactly glycerol, but glycerol type molecules. These molecules will then diffuse through the cytoplasm, and they can bind to receptors within the cell. If I were to give you an example of this last point, I will give you an example. Okay, let me give you an example of something that you have studied before, but not in this much detail. I'll just tell it to you like an example. Your endoplasmic reticulum. So in certain type of cells, for example, in pancreas, the endoplasmic reticulum also stores calcium ions. In muscles as well, the endoplasmic reticulum stores calcium ions. That is one of the functions. Of course, we have studied protein synthesis and packaging. It does that, but it also stores calcium ions. So what happens is that in your pancreas, the this phospholipid from the cell membrane is going to break down. It's then going to go through the cytoplasm. It will attach to the endoplasmic reticulum and it will release the calcium ions, right? And the calcium ions then help in release of digestive enzymes from the pancreas. That is one example. So these are the different roles that we have of phospholipids within our syllabus. Any confusion so far? Everything is making sense? Anybody has any questions?
No.
Aruj, all clear? Okay, guys, we're going to go ahead and then stop it over here for today. And inshallah, in the next class, we're going to go ahead and continue with our topic. Okay.